Hey everyone, today we're going to be wrapping up our lung physiology series with lecture 5. This is going to be slightly longer as we consolidate and expand on the concepts we've built across the unit. So as you can see, this is a pretty large lecture that I've intentionally divided into two parts. Both parts are required material. The content naturally fits together this way. Part 1 builds core concepts and kind of finalizes the unit for respiration. And part 2 is more of an integrative unit that, everything across multiple different systems. So the whole picture starts to make a lot more sense in PSL 301. Now, because this lecture works best as cohesive sequences, lecture 4 was intentionally kept shorter to balance the flow of the unit. I highly recommend taking a short break between the two parts to really help consolidate the ideas before we move to the integrated section. So let's start today with a real case study that highlights why neural control of breathing is so important. So this is Liam right here. He was an 11-year-old boy with a condition called congenital central hypoventilation syndrome, often referred to by the older name known as Ondine's curse. I'm not sure if you all heard about it. And it's named Ondine's curse because it's named after a German myth about a water nymph named Ondine who cursed her unfaithful husband to lose the ability to breathe automatically. Now, the condition, it describes congenital central hypoventilation syndrome. It's where a person cannot breathe automatically during sleep. And it was a state that essentially mirrors that curse. During the day, Liam breathes completely on his own. There's no problem. His diaphragm works, his respiratory muscles work, and he's awake enough that his voluntary and behavioral drives keep that ventilation going. But the moment he falls asleep, he loses his ability to have this automatic control of breathing. His brainstem simply doesn't generate the rhythmic drive to breathe during sleep. So what happens is that every night he needs to be connected to a ventilator through a tracheostomy tube just to stay alive. And so this condition tells us something incredibly important, that the automatic rhythm of breathing is not voluntary. It's not luxury, right? It's generated by a very specific neural circuits in the brainstem. And when those circuits fail, ventilation disappears, even though the lungs and muscles are completely fine. And so hopefully at the end of this lecture, you're going to be able to understand this a lot better. Before we get to the neural control, I think it's important for you to take a step back and recall the slide and all the important muscles that are involved in breathing in and out. Now, all of these respiratory muscles we previously talked about require a coordinated neural drive. They don't activate on their own. The brainstem has to rhythmically send signals down the spinal cord to drive inspiration and, when needed, expiration. And this slide hopefully will set the stage for understanding what the neural drive actually looks like. So the question you might be asking yourself is, who actually activates these respiratory muscles, and what is the final common pathway that every breathing command must go through? And so the answer is quite simple. It's the spinal motor neurons. All respiratory drive, whether it's voluntary control from the motor cortex, emotional inputs from the limbic system, or even the automatic rhythm generated in the brainstem, they must go through these spinal motor neurons before the muscles contract. So let me also tie this back into the spinal cord anatomy you might have learned in PSL 300, just to ground that structure into something familiar. And if you've not taken PSL 300, no problem, I'm going to explain it now. So motor neurons sit in the ventral horn right here in this butterfly shape right in the front of the spinal cord. And that's the interior portion of the gray matter, which is dedicated to output. Sensory afferents enter dorsally. Dorsal is right at the back here of your spinal cord. But that's not where breathing commands originate. Respiratory motor output always emerges from, remember, the ventral horn right here. And that's because that's where the lower motor neurons live. So for example, the diaphragm, the relevant group is going to be the phrenic motor nucleus. And those are located in the ventral horn segments of C3, C4, as well as C5. And these axons travel together to form the phrenic nerve, which then drives your diaphragm to contract. Now, for the intercostal muscles, the motor neurons are found in the ventral horn, of the thoracic spinal cord, and that's going to be from T1 down to T12. And these form the intercostal nerves, which activate the intercostal muscles to lift or depress the ribs during breathing. So the big takeaway is that all of the complex supraspinal control we'll talk about today, brainstem rhythms, chemoreceptive drive, voluntary override, they must funnel down into these ventral horn motor neurons, and they are the final common pathway for ventilatory control. So who tells those motor neurons when to fire rhythmically? And the answer really lies in the brainstem, specifically the medulla. So if you remember PSL 300, you remember the cardiorespiratory centers. And that's really the lower portion of the brainstem, the medulla. And just to ground you again on the anatomy, the brainstem has three major parts. We have the midbrain at the top, the pons, which is in the middle, and then the medulla. And that makes your brainstem. And this sits right above the spinal cord. And the medulla contains many of those vital autonomic centers that regulate the heartbeat, blood pressure, and breathing. Now, within the medulla, we have specialized populations of neurons called the premotor neurons. Premotor neurons are neurons that do not directly innervate the muscles themselves. Instead, they send those descending outputs to the motor neurons in the spinal cord. In other words, they are the command cells that shape the timing and pattern of that motor neuron activity. For breathing, these motor neurons generate the automatic drive or rhythmic drive, and they're the ones that say breathe in, breathe out, breathe in, breathe out all day long and all night long. And you could see that in the diagram here, the medullary premotor neurons at the top sending a descending projection into the cervical spinal cord where they synapse onto the phrenic motor neurons in the C3 to C5 region, and the phrenic nerve carries that signal directly to the diaphragm. So hopefully that makes sense here. On this slide, we're looking at one of the most important functional relationships in respiratory physiology of how neural circuitry activity of the inspiratory motor circuits translate into changes in the tidal volume, that quiet breathing. So on the top graph that you see right here, that red line, that sinusoidal wave, really represents tidal volume, how much air is moving in and out of the lungs with each breath. And you can see this normal breathing cycle. Inspiration rises over about 2 seconds, and then expiration falls over about 2 to 3 seconds. Now, look at the lower graph right here. This beautiful blue, spiky looking graph. This is showing you the number of active inspiratory neurons over a period of time. Specifically, the premotor neurons in the medulla that eventually drive the motor neurons for the diaphragms, which eventually contract. And this lower blue trace represents what's essentially what we call the phrenic nerve activity pattern. And so you're seeing essentially the neural drive and not the airflow. So during the inspiratory phase, there's this characteristic ramp up pattern at the start of inspiration, and only a small number of inspiratory neurons are active. But as inspiration continues, more and more of those neurons become recruited, and that's what that green arrow is really highlighting right here. Okay, and the more inspiratory neurons that fire, the more additional neurons join in, which progressively increases the strength of the inspiratory drive. And this is why the phrenic nerve activity ramps up instead of turning all at once. And as the neural drive increases, the diaphragm contracts more strongly and the tidal volume increases in parallel. So the top and bottom graphs are tightly linked. More inspiratory neuron activity gives you a deeper breath. Eventually, we reach to the point where inspiration has to stop. And this is marked by that red arrow right here. And inspiration shuts off, and that's an intrinsic medullary mechanism, often involving inhibitory circuits in the pontine respiratory group. We'll talk about the PRGs, and the stretch receptor feedbacks, again, we'll talk about it, that abruptly terminates that inspiratory activity. And you'll notice the blue trace kind of drops so dramatically, right? Like as if you're in a roller coaster. And the diaphragm then relaxes and the tidal volume begins to fall, making the start of that passive expiration. Expiration here is passive. Remember, notice that there is no ramping pattern. So the number of active inspiratory neurons just stay low, very low baseline during this time because they're essentially silent. The lungs recoil elasticity brings back, the lungs recoil elastically brings it back to their resting position. Then the entire cycle just repeats again. You get inspiration, expiration, and so on and so forth. The figure on the left shows you key structures in the pons and medulla, and we're going to walk through one by one. So starting from the medulla, which is right here at the bottom, we have two major groups. You have the DRGs, which is the dorsal respiratory group, and it just sits in and around the nucleus tractus solitarius. You might have heard about the NTS, which is the major relay site for sensory input coming in through cranial nerves number 9 and number 10. And this includes information from lung stretch receptors, airway mechanoreceptors, as well as arterial chemoreceptors. Now, because of that, the DRG is heavily involved in integrating sensory feedback, okay, from the NTS especially. And it's going to shape the basic inspiratory pattern. The DRG primarily contains the inspiratory neurons, which send their output to those spinal motor neurons that control the diaphragm. A quick way to remember this is dorsal detects. So dorsal DRG, and it's associated with the sensory detection through the NTS. Now we also have what we call the VRGs, or the ventral respiratory group. And so the VRG is a little bit more complex. It contains both the inspiratory and the expiratory premotor neurons. And it becomes especially important during active or forced breathing, for example, during exercise, speech, or cough. Within the VRG, we have what we call the pre-Butzinger complex. Okay, and that's written right here. It's a really interesting name, which many studies suggest it's the critical rhythm generator. And think of this region as essentially containing those pacemaker-like circuits that then set the timing of breathing. A useful memory cue might be ventral for volume. So VRG ramps up air movement when ventilation needs to increase speaking, coughing, or exercise. So in the medulla, rhythm emerges from the pre-Butzinger area and is shaped by the DRG and the VRG before descending to the spinal cord. Now, just above the medulla, we have the pons, and this is this region right here. And this is called the pontine respiratory group, or the PRGs. And the PRG receives the input from the higher brain centers, including behavioral and emotional pathways. For example, the limbic system. Its main function is to fine-tune the rhythm, particularly to help terminate inspiration and regulate the transition between inspiration and expiration. And this is why damage to the pons can produce very long gasping breaths, because the normal off switch for inspiration is now disrupted. So pons, I like to think of pause. Pons, pause. And so PRG helps pause inspiration and switch to expiration. So let's put this together. DRG integrates sensory feedback and generates a basic inspiratory pattern. VRG, including the pre-Butzinger complex, provides that rhythm-generating circuits and contributes to both the inspiration and expiration. And PRG is the pons adjusting timing, ensuring that breaths are smooth and that they're appropriately phased. And the NTS that you see right here, it's the major sensory relay that allows the medulla to adapt breathing to mechanical and chemical stimuli. Memory anchor for NTS might be the node of traffic signals. So all sensory traffic kind of comes in here. Now, all of these groups contain premotor neurons, which then project down to the spinal motor neurons, like the phrenic and the intercostal motor neurons we talked about previously, to drive those respiratory muscles. So the respiratory rhythm isn't generated by a single spot. It arises from a coordinated network across the medulla and pons that continuously integrate sensory input, behavioral demands, as well as metabolic needs. So now that we've seen the broader layout of the DRG, the VRG, the PRGs, as well as the NTS, we're going to kind of zoom in to the specific medullary circuitry that actually produces the respiratory rhythm. Everything on this slide represents the core microcircuit that you see right here, that generates and also shapes the 3 phases of breathing cycle that we see in normal eupneic breathing. And the 2 key regions we're focusing on are the pre-Butzinger complex, which is right here, or also known as the pre-Botzi, and then the Butzinger complex, which is Botzi. And these are neighboring structures in the ventrolateral medulla, and together they form the foundation of the respiratory rhythm as well as the pattern generator. And the pre-Botzi is shown on the right side of the top diagram right here, and this is the Botzi on the left side. So before we start talking through the nitty-gritty details on the circuitry, let's anchor ourselves in the phases of normal breathing, because the traces at the bottom here that you see correspond to a lot of those phases. So inspiration is when the diaphragm contracts and the upper airways open, and post-inspiration, which is this part right here, is a part of expiration, and that's the early part of expiration. So it's where airflow kind of slows down a little bit. And then you have your late phase of expiration, which is mostly passive at rest, but becomes active during exercise. So there's really three phases: inspiration, post-inspiration, which is also the early phase of expiration, and then the final stage of expiration. Okay, so now let's take these phases and map them onto the neural populations that are shown in this top diagram here. So the starting point of every breath is the pre-Botzinger complex, so right here. Inside the dashed outline here in the top figure, the pre-Botzi really contains a small population of excitatory glutamatergic neurons that express a special current, and that's written right here. Okay, so that's known as the persistent sodium current. The INAP is what I like to call it. So these persistent sodium channels don't behave like the fast spiking generating sodium channels that you see in action potentials. Instead of opening and closing rapidly, they open partially and they stay open for a longer period of time, producing these slow inward sodium leaks. And that slow depolarizing current allows these pre-Botzi neurons to burst rhythmically on their own, even when isolated in a slice. Now, this intrinsic bursting behavior makes them what we call the kernel, which is what we wrote here, the kernel of the respiratory system. In other words, the pre-Botzi really determines when each inspiratory cycle begins. Okay, so if you look at the bottom panel again, the first trace you're going to see is the pre-I population, which is right here, okay, and it's in red, and it's kind of color coordinated as well. So these neurons, what you'll notice is that they start firing slightly before inspiration begins, right here. Okay. And their early activity firing is actually really important because it's going to activate the upper airway dilator muscles. For example, the genioglossus muscle of the tongue via the hypoglossal nerve, cranial nerve number 12, to stiffen and open the pharyngeal airways. Now, this is important because when the diaphragm contracts, it generates this negative pressure that we talked about that would otherwise tend to collapse the pharynx. So the pre-I neurons act like the system's airway preparation signal, and they open the gate before air actually flows in. And so that's why it's really important to have that little preemptive firing before the inspiration happens. So next is the early I neurons that activate, and you can see that their sharp burst at the beginning of the white region happens right here. So this is the early, and it's right here. And this is to sharpen the inspiratory command that's coming from the pre-Botzi here, and they from this pre-I, and they contribute to turning the pre-inspiratory activity into more of a robust inspiratory motor burst. Now, finally, you also have what we call your ramp I neurons, or your ramp inspiratory neurons, and they're located in the rostral VRG, right here. And these are the firing patterns that you see right here. Okay, so these neurons are premotor neurons that project directly to the phrenic motor nucleus in the cervical spinal cord, and their slow upward ramp that you see right here during inspiration, okay, that ramp creates the rising slope of the phrenic nerve activity, and that produces that smooth graded activation of the diaphragm. Okay, so the inspiratory sequence in the bottom diagram works like this. You got the pre-I/I inspiratory that prepares the airways. You get the early I that triggers inspiration, and you get the ramp I that also shapes the inspiratory motor output. Okay, so now we're going to move on to this left side, which is the Botzinger complex, or the Botzi. And unlike the pre-Botzi, which is essentially dominated by excitatory inspiratory neurons, the Botzi contains mainly inhibitory expiratory phase neurons that you see right here. And so the two major groups that I want you to memorize and know are the post-I neurons right here, post-inspiratory, and the augmenting expiratory, or the Augie neurons right here. Okay, so if you look at the post-I trace, which is this first one right here, you'll see that these neurons become active immediately when inspiration ends. Post-inspiration, says it in its name. And so their job is to essentially terminate inspiration and begin the post-inspiratory phase, or what we call E1, the early phase. And it's one of their most important actions in activating what we call the laryngeal adductor muscles. And these are the muscles of the vocal cords that narrow the glottis. Okay, so you might want to Google what a glottis is, and you can kind of see how it looks. And this partial glottic narrowing acts as essentially what we like to call an expiratory braking. And it essentially what it does is it's going to slow the airflow leaving the lungs during an expiration. And by doing that and creating a bit of resistance, it maintains the end expiratory lung volume, and it prevents the small airways from collapsing. Because if you didn't have a bit of resistance and everything was just open, your lungs would just kind of shut with all that pressure going out. Okay, so it's going to prevent that small airways from collapsing. So it's not a full glottic closure. It is a gentle narrowing to control the rate at which air leaves the lungs. So that's also important. Now we also have what we call the AUG, which is the augmenting expiratory neurons. And so lower down here, which is what I'm showing you here, the AUG-E neurons show a more gradually increasing firing pattern during the second phase of expiration, which is called the E2 phase. And you can see that right here. They're not active here, they're active here. At rest, this activity is low and expiration is mostly passive elastic recoil, as I told you multiple times. But during exercise or increased ventilatory demand, it could be that the AUG neuron, and it usually is, where the activity starts to strengthen. You're going to see their activity go up. And it helps recruit that abdominal and internal intercostal muscles for that active expiration that you need. So now that we've seen essentially the inspiratory and the expiratory populations here, now if you kind of look again, right, the key principle is that it organizes them into the cycle of mutual inhibition. So if you look again at the top figure here, the blue inhibitory synapses, they create a ring-like circuit. So during inspiration, the pre-I/I and the early I neurons, they inhibit the post-I and the AUG neurons. During post-inspiration, the post-I neurons inhibit all the inspiratory groups. And then during the late inspiration or expiration, the AUG neurons will inhibit both the post-I and the inspiratory neurons. So this mutual inhibition forces the system to move through the breathing cycle only in one direction: inspiration, post-inspiration or E1, and then E2, your late expiration. And so this entire inhibitory network is what we call the pattern generator. It determines the sequence and the shape of each breath. Now, before we end the slide, you'll notice that there's other neurons that do play a role here, and these are tonic drives that feed into this network from, let's say, the pons, which are right here, the RTN right here, and then the raphe nucleus. And so these inputs modulate excitability and allow the rhythm to really adapt to the behavioral state and the chemical stimuli. So what the pons do is that they're going to smooth the phase transitions, and they help maintain that normal inspiratory-expiratory balance. So kind of like this nice wave. The raphe nucleus that you see here, the nuclei here, provides that serotonergic input that stabilizes breathing and is particularly important during wakefulness. And you might have heard this in PSL 300 as one of its roles. So this should be a review. And then you have the RTN, which is which is right here, also contributing to this tonic drive. And it's highly CO2 sensitive. And this increases its excitatory drive when PCO2 rises. And this increases the respiratory frequency as well as their depth. Okay, so now you should be able to comfortably explain what the slide's going on and everything I just said. And feel pretty comfortable with all these terminologies. Everything here is fair game. And I can definitely ask you anything from here. So I forgot to mention on the previous slide, but you should know the full name for RTN, which is the retrotrapezoid nucleus. And it could be asked either ways on the exam. So you should feel comfortable with the short form, but also the long form. Now this slide here shows two sets of traces. You have panel A is really about an in vivo model. And this activity is recorded from living animals' respiratory muscles, and panel B is an in vitro medulla slice, where the activity is recorded from an isolated medullary slice that contains that pre-Botzinger complex that I was talking about. So in eupnea, which is normal breathing, the in vivo eupnea is that regular quiet breathing rhythm. And this waveform has an augmented bell-shaped profile, and it starts small and rises steadily and then falls smoothly. And you can see that here. Okay. In the isolated pre-Botzi slice, which is right here, you still get that same bell-shaped pattern. Okay, and so even though the lungs and muscles and higher brain centers are gone, they're still firing very similarly. And so what this tells us is that the shape of the inspiratory burst, its gradual rise and then a fall, is generated intrinsically by the pre-Botzi and its surrounding VRG pattern generating network. Another concept that I want you to see is something called size. And size are these large amplitude pretty complex bursts that occur periodically during normal breathing in vivo, and you can kind of see that right here. Okay, and this happens again during in vivo, and this serves a physiological purpose, such as reopening, let's say, a collapsed alveoli, so it's going to recruit it, and resetting that respiratory patterns. And then during the initial phase of hypoxia, which is shown in panel B, you get that psi frequency that increases, and this is known as augmentation. And so what's remarkable is that even in the isolated slice, the pre-Botzi also produces that fictive psi with a larger amplitude, complex shape, and slower frequency relative to eupnea. So the pre-Botzinger complex contains the circuitry required to generate psis independently of the peripheral sensory input. So it's pretty nice. Now, if you take a look, we're going to talk also about gasping. So when hypoxia becomes more severe, the respiratory pattern switches from eupnea into gasping, which has a very fast rise time. And you can see that right here. Okay, and then brief, sharp bursts, and then a simple spike-like form that you see like this. And so this transition is known as the late phase of hypoxia or hypoxic depression. And again, this pattern is reproduced almost perfectly if you kind of take a look at this fictive gasping with the isolated pre-Botzi slice. And the sharp, fast rising waveform really shows that when network excitability is very low, for example, as in hypoxia, the intrinsic pacemaker properties of the pre-Botzi neurons dominate, producing that simplified survival rhythm. And so this is why gasping is thought to be kind of that last resort brainstem rhythm that persists during severe metabolic distress. Okay, and then panel D is just kind of overlapping everything to show that it's all very similar. So in both in vivo and in vitro recordings, when you overlay the three rhythms, you have the eupnea in black, psi in red, and gasping in blue. What you see again is this eupnea, nice, smooth, bell-shaped. You see that large complex burst from sighing, and then you also see that sharp, fast rise time burst from gasping. And this demonstrates that all three rhythms emerge from the same pre-Botzi network, but their expression really depends on the network configuration and those neuromodulatory states. Now, hypoxia does not simply weaken the network, it just reconfigures it by shifting the balance between excitation and inhibition, and the contribution of the intrinsic pacemaker currents, the level of tonic drive of the RTN, and the RAFE and PONS. And so the pre-Botzi is not just a pacemaker, it's a kind of a multifunctional, state-dependent rhythm generator. And so what this experiment I think would show you is that the pre-Botzi complex is sufficient to generate multiple breathing rhythms. Breathing rhythms are not hardwired. They are dynamically reconfigured based on metabolic conditions. Hypoxia rapidly changes the breathing patterns from eupnea to sighing to gasping as the network transitions through different excitability states. And even in isolation, the pre-Botzi produces these rhythms, meaning these behaviors are embedded in its microcircuitry and neuromodulatory architecture. And so this is one of the strongest lines of evidence, and this is why we show it to you in this course, that this pre-Botzi complex is the core rhythm generator for breathing capable of sustaining life, you and I, and sustaining these life-preserving patterns even when the rest of the brain, for example, is compromised. So now that we've looked closely at the rhythm generator in the medulla, especially the pre-Botzinger complex, we need to talk about the structure that helps shape that rhythm so that breathing is much more smooth and well coordinated. And I kind of alluded to this already, but this is the pontine respiratory group, or the PRGs, that are located in the pons above the medulla. And now, so the PRG is not a rhythm generator itself. Instead, its role is entirely modulatory. It receives sensory information from the DRG. Remember, the DRG sits around the NTS, and it's integrating inputs from like lung stretch receptors, airway mechanoreceptors, and chemoreceptors. And once that sensory information reaches the pons, the PRG uses it to help fine-tune the timing of inspiration. One of the most important things of the PRG does is to control the termination of inspiration. In other words, it helps determine when the medulla should shut off that inspiratory burst. Because without the pontine influence, inspiration tends to run long, producing the slow, gasping-like breath. And this is exactly what we see, for example, in experiments where the pons are removed, breaths become more prolonged because the medulla doesn't receive that normal off-switch signal, right? So in addition to processing sensory information, the PRG also provides that tonic excitatory input to the medullary respiratory circuits. And this tonic drive, if you look at the circuit, you'll see it, helps stabilize the rhythm and prevents abrupt transitions between that inspiration and expiration. So it's essentially helping to keep the entire system balanced so that breathing remains smooth, coordinated, and also appropriately paced. So now that we built the core framework for how the medulla and the pons generate and shape the respiratory rhythm, it's really important to remember that the rhythm is highly modifiable. So breathing is not a rigid process or a metronomic process. People do think like that, but it's not true. It's constantly being shaped by multiple inputs acting on the central nervous system. So on this slide, what I've done is I kind of listed out a range of factors that can influence rhythmic breathing. So first, we have what we call voluntary control right here. Okay, speech, exactly what I'm doing now, singing, which I'm definitely not doing, or even breath holding. So these behaviors can originate, let's say, from the motor cortex, the higher order centers, and descend through the corticospinal tracts to override some of that automatic brainstem rhythm. And this is why you can hold your breath, for example, even when your chemoreceptors are telling you to breathe, right? At least for a while, obviously. And then we also have, for example, the chemoreceptors, which are right here. And they provide kind of that chemical control of breathing. So we talked last class about central chemoreceptors in the medulla and how they respond to changes in the CO2 and the pH, while the peripheral in the carotid, for example, and the aortic bodies respond to also CO2, but they can be augmented by oxygen. And so these inputs feed into the NTS that I talked about and directly modulate the rhythm generator, often quite rapidly. So there are ways to modulate breathing. We also have reflexes like sneezing and coughing, and these are hardwired protective reflexes that are driven by sensory receptors in the airways, and when triggered, they override the normal respiratory rhythm to expel potential irritants very forcefully out. Posture can matter as well, so if you slouch, or if you compress your abdominal cavity, it mechanically alters the intercostal and abdominal muscles, and that can affect lung volumes and the ease of inspiration, if you will. And so the brainstem rhythm stays the same, but the mechanics change. So the central nervous system kind of has to adjust a little bit about the neural drive accordingly. You might get what we call a startle response, which can produce abrupt increases in breathing via very rapid activation of the brainstem and limbic circuits. And this is why, when someone surprises you, your breathing reflexively spikes and you're like, right? And then we also have emotions, after somebody, scares you, all the collateral damage that happens after it, right? So the fear, the anxiety, even maybe potential sorrow. And so these are processed through the limbic system, the hypothalamus and the cortical centers, all of which send those descending projections to those respiratory networks. And this is why anxiety disorders are often, they have this physiological presentation or manifestation of what we call and see hyperventilation. So a lot of these patients have that. And that's why crying kind of at times disrupts those breathing patterns. Exercise is another really good example. So it increases ventilation immediately, even before CO2 levels rise, through the central command signals from the motor cortex and mechanoreceptors in the muscles and joints. And so this is an example of what we call a feed-forward control. And finally, you also have pain here, which can increase breathing rate through strong nociceptive, which are your pain signal inputs, projecting into the brainstem and the hypothalamus. Again, this all overrides the baseline rhythm, right, to match that physiological stress response. So the key point of all these influences, they converge on the central nervous system. The medulla and the pons generate the rhythm. The medulla generates the rhythm, but the pons kind of refine it. But the rhythm is constantly being shaped by voluntary input. Reflexes, emotional states, sensory feedback, and behavioral demands. And I think this is really what's important. This is what allows breathing to be automatic, but also highly flexible. So this slide here takes all those factors that I talked about in the previous slide and puts them into system level diagrams so that you can see how they all converge on a respiratory network. So if you start on the left side at the top, you're going to see the voluntary control coming from the motor cortex. And so this includes things like speech, singing, breath holding, and any deliberate changes in your breathing pattern. And so these are cortical signals that bypass the brainstem rhythm generator and project directly to the spinal respiratory motor neurons. That's how you can consciously override your automatic rhythm. Right below that, you see coughing and sneezing, which originate in the medulla. And these right here, these are more reflexive protective behaviors that are triggered by sensory input from the airways, and they also send strong descending commands directly to the respiratory motor neurons. During a cough, for example, the brainstem essentially hijacks the breathing circuit to produce a massive coordinated expiratory burst. Moving slightly to the right is posture in the cerebellum, which can also affect breathing. This is because certain postural configurations can change the mechanical advantage of the intercostal and abdominal muscles. And so the cerebellum's adjustments project both to the rhythm generator in the medulla and directly to the spinal respiratory motor neurons. Now in the middle, we have the rhythm generator, which is right here, the medulla. And so the medullary networks that you just learned from me now, the pre-Botzinger complex, the Botzinger complex, and the VRGs, all part of the VRGs, these circuits continually generate that baseline respiratory rhythm, and everything else in this diagram is helping to modulate or override them. And so on the right, if you take a look here, okay, you can see influences from the forebrain, specifically, emotions like fear, anxiety, sorrow. These emotional states are processed through what we call the limbic system, which then projects to the reticular formation, which is a widespread modulatory network in the brainstem. And from there, those signals can either modify the rhythm generator or act directly on the respiratory motor neurons that are shown right here. And finally, at the bottom right that you see here, so that's to do with sensory stimuli. I spent a whole lectures in PSL 300 talking about sensations, but sensations such as pain or startle responses, these signals feed into the reticular formation as well, and they can produce this rapid increase in the respiratory drive. And this is why, for example, pain or sudden loud sounds instantly can speed up your breathing before CO2 levels changes. And so what the diagram here really is showing you is that all of these diverse inputs ultimately converge onto two output nodes. You have the medullary network here, okay, that can adjust the timing of breaths, or you can just go straight directly to your spinal respiratory motor neurons, and that can execute that actual motor pattern. So all these can go straight directly down, and your reticular formation can go to modulate your medulla or your spinal cord. Okay, so this slide brings everything together by showing how those influences actually reach the diaphragm, which is a major organ to allow you to inspire air. In other words, this is the final efferent pathway that drives inspiration. So I'm going to show you and kind of orient you from this diagram. So at the top, at the bottom here, what you see is the diaphragm, and immediately above it is the phrenic nerve, which remember impinges on the diaphragm, which originates from the phrenic motor neurons that are located in the cervical spinal cord from the segments C3 to C5. And these spinal motor neurons are the final common pathway for the diaphragm activation. No matter where the signal starts, cortex, brainstem, limbic system, it must ultimately activate these particular neurons. Now, I kind of want you to follow what I kind of highlighted in like thick black arrows and go upwards. So that projection really represents the autonomic or the involuntary control, the automatic control that's coming from the brainstem here. And so this is the pathway that's generated by the pre-Botze complex. And specifically, it's within the VRG group. And the whole medullary pattern generator we've now seen in multiple diagrams. And so this pathway ensures that your diaphragm contracts rhythmically without you needing to even think about it. Okay, so that's this pathway. Now, I want you to look at that dashed vertical line that's coming from the cerebral cortex, which is right here. And this represents the voluntary control. So previously I just talked about was the involuntary. This is the voluntary control. This originates from the primary motor cortex and the premotor cortex, which you see right here. And this is how you hold your breath, how you speak, like what I'm doing now, how you sing, how you blow out a candle. Any deliberate changes in your respiratory pattern, it's coming from these, from the cortices. So what's important here is that the cortical pathway now descends directly to the spinal phrenic motor neurons, bypassing that brainstem rhythm generator, and that's why you can override your automatic breathing temporarily. But as we discussed earlier, the moment your CO2 rises enough, the chemoreceptor input will then force the brainstem rhythm to kind of reassert control, and then you involuntarily take over your breath. Finally, notice the branching arrow labeled as brainstem. So this, essentially, this part here, okay, this shows you that medullary respiratory network not only generates rhythm, but can send that descending output through the spinal cord to the diaphragm, and it's also the location where those reflexive behaviors like coughing, sneezing, and gasping originate from. And I talked about them in a couple slides before. So hopefully this kind of just puts everything together, and you get now a visual diagram. And when you thought you didn't have enough, you have more. Welcome to physiology. Okay, so now that we've gone through how the rhythm is generated in the medulla and how the pons help shape it, this slide kind of brings everything again together. So breathing is not just a one-way motor command, as I told you. It's a closed loop system with continuous communication between the brainstem, the muscles, and the sensory receptors. So first, we're going to start with the efferent pathway. This is the outgoing motor command. So we're going to take a look at the red. Okay. So what you see here are the efferent signals originating from the medullary respiratory centers. Do you see that right here? Efferent. Okay, so these include the pre-Botzinger complex, the Botzinger complex, and the general VRGs and the DRGs. And these neurons send commands to your spinal respiratory motor neurons, which activate the diaphragm and intercostal muscles. Okay. So those are your respiratory muscles. And it also activates your upper airway muscles, which, of course, right here are important. I told you about keeping them open during inspiration. And we also have your airway smooth muscles. It's mostly reflexive and it's controlled for that bronchodilation and constriction. So that's all controlled by the vagus nerve. And so all of these outputs together determine the pattern of pulmonary ventilation. Now, I want you to follow the feedback circuit into the brainstem. So once the muscles contract and air actually moves in, the sensory receptors return that information back to the brainstem to confirm that there's air inside. And that's shown here in the green pathway, and that's known as the afferent pathway when signals come back into your integrating centers. So that's the afferent pathway. And so what we have here is we have quite a few things going on. The central chemoreceptors, which are right here in the medulla. They sense CO2 changes in the protons in the environment of your CSF, and they send those powerful feedbacks to the medulla, respiratory neurons, and this is our dominant long-term controller for breathing. We talked in a previous lecture about the peripheral chemoreceptors, remember the carotid bodies, and so those respond rapidly to low PO2, high H plus, and CO2. And so they're going to send their signals mainly through cranial nerve number 9, and that's what you see right here. And that's known as the glossopharyngeal nerve. And so these are important in, for example, situations of acute hypoxia, maybe in altitude or breath holding, airway obstruction. So they do play a big role here. Another one I want you to know is, and I'll talk a little bit more extensively about this later on, it's about the lung and airway stretch receptors, and it'll be also through vagus nerve number 10. Okay. So again, this vagus nerve number 10 was the efferent pathway. This one is about the afferent pathway. And so these include those slow adapting stretch receptors that participate in the Herring-Brewer reflex, which I'll talk about. And they essentially just tell the medulla whether the lungs are inflated and whether air flow is occurring, and whether irritants or excessive stretching are present. And so they need that feedback to make some decision on what to do with the breathing. So together, all these green arrows represent this continuous sensory correction of the rhythm. Okay, and you'll notice something a little strange here are these muscle receptors. Okay, so these are a separate branch of feedback from the muscles themselves. So these might be muscle spindles and tendon organs, and essentially these help the medulla match the intended motor output to the actual mechanical performance. This is important. For example, if you're exercising, if your posture changes or there's some sort of resistance to breathing, you do need some level of muscle feedback. And that's what this is showing you here. So what's the big picture here? The medulla generates the rhythm, the efferent motor output drives the muscles, and the lung and the environment changes accordingly. Chemoreceptors and mechanoreceptors will send that information back, and the medulla adjusts the rhythm in real time. And so this closed loop control system is why breathing can instantly and adjust when you start exercising. For example, when CO2 rises, when your posture changes, or when the airway resistance increases, your breathing is able to adapt. So I just alluded to this concept in the previous slide. Now we're going to move from central rhythm generation network back to the periphery. And we're going to talk more about it from the perspective of sensory receptors that are inside your airways and lungs that feed information back to that brainstem. And these are the stretch receptors and irritant receptors that I was talking about. So these receptors essentially provide those critical afferent feedback that helps the medulla fine-tune your breathing pattern and detect irritants and prevent overinflation of the lungs. So we're going to walk through 3 major receptor types that I want you to know. You have the stretch receptors, the mechanoreceptors in the airways, and you'll notice there's 2 kinds of, on the left, we have what we call the slow adapting receptors, and you have the rapidly adapting receptors. It's actually much more intuitive than you think. So they're both mechanoreceptors, meaning that they respond to changes in lung inflation, but the SARS, or the slow adapting receptors, they respond and they fire continuously along as the lung is inflated. And you can see that here, right? This is your lung volume as it's going up. They're firing throughout as long as the lung is inflated. And again, you can see that trace going up. Now, these receptors are part of the classic Herring-Brewer reflex, which helps terminate inspiration when the lungs stretch enough. We also have the RARs, which are the rapidly adapting receptors, and they're different. So if you look at the second trace right here, they definitely look different. And they fire a huge burst at the beginning of inflation, at the beginning of inflation, and then quickly they'll quiet down. And you can kind of see that right here. They don't fire much anymore. And so these receptors are more sensitive to sudden changes, like the phasic response that I was talking about in PSL 300, like when the airway collapses or when there's a mucus or dust. So they're really known to rapidly respond to these fine changes. And so they might trigger something like coughing or rapid shallow breathing. That's where RARs comes in. Now, both SARS and RARs, they send that information to the brainstem through those myelinated A fibers, okay? And they will conduct signals pretty quickly. Now, we also have the irritant receptors. And on the right of the slide, you can see these are the C fiber endings. And as you can see, all your PSL 300 is coming back. No, you don't really need it to understand this, but it feels great when you know a little bit about it, right? So you can see those C fiber receptors. Some textbooks might say J receptor. We're not doing that. We're just going to call it C fibers. And so these are unmyelinated fibers, so they conduct a lot more slowly. So remember we talked about cable mechanics, but importantly, they're highly sensitive to chemical signals. Things like capsaicin, histamine, bradykinin, all stuff that you probably learned at the beginning of the course, smoke, and inflammatory mediators. And if you look at the recording on the right here, a small injection of capsaicin causes that immediate burst of activity in your C fiber trace, and it's followed by changes in the tracheal pressure. Okay. And you can see that right here. And that when activated, these receptors trigger that coughing, airway constriction, and sometimes apnea that's followed by that rapid breathing. So these are kind of your airway danger sensors, if you will. So why does this matter? Well, all these three receptors, they send their feedback to the medulla, and specifically the NTS that I talked about. So the brainstem can kind of adjust the breathing in real time. And this is how your respiratory system prevents that overinflation, and it responds to those irritants and changing patterns during diseases and exercises. So these receptors that I was talking about, the stretch receptors, they're found throughout the trachea, the bronchi, the bronchioles, and they're classic mechanoreceptors. And they respond whenever the lung tissue expands during inspiration. All of their sensory information travels back to the brainstem through the vagus nerve, and that's written here. Okay, and so this is another example of vagal afferent feedback that's shaping our breathing pattern. And so in animals, stretch receptors participate in a well-known reflex that I just talked about, which is the Herring-Brewer inflation reflex. And so when the lungs become very inflated, these receptors will fire strongly, and they'll send those signals to the medulla that essentially says, that's enough, stop inhaling. And this reflex prolongs that expiration and prevents that overdistension. Now, in adult humans, this reflex is much less dominant during normal and quiet breathing. We don't rely on it every breath. Instead, its role seems more subtle. It contributes to more fine-tuning the tidal volume and breathing frequency, especially during exercise, deep breathing, and in situations where lung volume might be changing a little bit more dramatically. So overall, stretch receptors are part of the system that helps maintain, or it helps the brainstem keep the breathing smooth and coordinated, and they modulate how big each breath is and how quickly each breath follows the next. Now we're going to shift from stretch receptors to irritant receptors that I was talking about. So these receptors are found throughout the entire pathway, airways, from the nose all the way down to your bronchi, and their primary job essentially is to detect anything that shouldn't be there. It might be smoke, dust, cold air, chemicals, allergens, and maybe other mechanical irritations. And when they activate, they send a very fast afferent signal, mainly through the trigeminal nerve and the glossopharyngeal nerve or the vagus nerve, depending on where the irritation actually occurs. And their activation produces two major responses. You get that immediate change in breathing, that irritant receptors can cause that rapid shallow breathing or even a brief apnea followed by that strong inspiration. And this is the airway trying to protect itself. So those reflexes are there to clear the airway and protect the lungs. And these include sneezing, coughing, as I mentioned. But if you look at the table below that I'm showing you here, you can actually see exactly which receptors trigger, for example, which particular cranial nerve. So for example, the nasal irritation would be your sneeze reflex through your trigeminal nerve, your laryngeal irritation, so cough will be through your vagus nerve, and the juxtacapillary receptors, or your J receptors, that are found deep in the alveoli. Those reflex changes would be in breathing. So it'll be your vagus nerve here. So clinically, these irritant pathways are extremely important. They're the reason you cough when you inhale pepper spray, and why cold air can actually trigger asthma, and why aspiration into the larynx immediately results in that forceful coughing. So before we dive deeper into the chemoreception and the neural circuits controlling breathing, I do want to show you the real clinical example of what happens when these systems don't develop properly. So I talked a little bit about this condition, CCHS. It's a rare disorder, and it's really caused by a mutation called the FOX2B gene, which is a transcription factor that's required for development of the autonomic neurons, including many of the neurons that provide an automatic drive to breathe. And because these circuits don't form normally, children with CCHS do not mount the usual ventilatory response to rising CO2 or falling O2, especially during sleep. They breathe, but the automatic control of breathing is quite impaired, meaning they can hypoventilate without ever feeling shortness of breath or feeling short of breath. So this syndrome is an important reminder that breathing depends on a very specific developmental program within the brainstem. And now in the next few slides, we're going to look at exactly which chemoreceptors and brainstem structures normally provide this automatic drive. And this is how defects in the FOX2B can disrupt the system. Okay, so again, before we go deeper, you should recall how chemoreceptors regulate. This is just a quick review. So what you're looking at is the expression pattern of the FOX2B gene in the mouse brainstem. Remember, FOX2B is the gene that defines CCHS, and it encodes the transcription factor that is absolutely essential for the development of those autonomic neurons, including the ones that are involved in breathing, chemoreception, and those broader autonomic regulation. Now, if you look at the figure here, FOX2B expression, it's highlighted through different colors based on their density. FOX2B expression you can see here, and they're especially dense in what we call the parafacial respiratory group, so more ventrolateral, this part here. Okay, and this is also called, this region that I'm pointing to right here is also called the RTN, or the retrotrapezoid nucleus. And this is one of the major sites for central chemoreceptions. And these neurons, I told you before, they detect increases in CO2 and the resulting increases in protons. And that provides the tonic chemical drive that keeps you breathing, especially during sleep. Now, the RTN is also receiving information from the peripheral chemoreceptors. So it all functions like an integrative hub for CO2, pH, and O2 as well. Now, here's where the clinical story all kind of connects together. So in CCHS, the FOX2B mutation, usually it's an expansion of the polyalanine repeat. This disrupts the development or the function of these FOX2B dependent neurons. And when these RTN neurons that you see right here loses its automatic ventilatory response to hypercapnia and hypoxia, that's why we see these children show those alveolar hypoventilation, especially during sleep. In more severe cases, even during wakefulness. So, but FOX2B doesn't only affect the RTN, just keep that in mind. So there's been more studies that came out, MRI studies and post-mortem analysis in these patients who died, unfortunately, where we can see these abnormalities across multiple autonomic respiratory related regions, including the hypothalamus, which contributes to thermal drive to breathing, the posterior thalamus, the midbrain, which all help coordinate the oxygen-related and rhythmic motor patterns. We talked about the raphe, right, the caudal raphe. So all these are really important for those serotonergic and modulation, for examples. The lateral medulla, the pons, and even the cerebellum. So tons of areas that are affected. So some areas of the cortex, even the insular and the cingulate regions, which normally contribute to the perception of dyspnea, they're also affected. So this explains why patients have altered thermoregulation, autonomic dysfunction, and absence of the perception of the shortness of breath, even though, for example, their voluntary breathing remains intact. So during the day, they look normal. So this slide essentially highlights the developmental dependency of the respiratory control network. If FOX2B dependent neurons fail to form to function, especially those in the RTN, the body simply cannot mount the automatic chemical drive to breathe. And this is why CCHS represents such a powerful model for understanding how the autonomic nervous system and respiratory networks are wired together. So before we wrap up this particular section on respiratory control, I do want to show you the real example of how this knowledge translates into treatment, how physiology can translate to the bedside. And this slide basically shows you a phrenic nerve pacer right here. And this is one of those main therapies used for individuals with CCHS. So the brainstem member doesn't automatically drive breathing during sleep. The diaphragm can work, but the rhythm generator doesn't activate. So a phrenic nerve pacemaker can bypass that missing brainstem signal. And so if you look at the image here on the right, you can see the small electrodes. Okay, these are the small electrodes that are surgically placed around the phrenic nerves. And then a receiver is implanted under the skin, and an external transmitter sends the electrical pulses wirelessly to the receiver, and each pulse stimulates the phrenic nerve, which then contracts the diaphragm and produces a breath. So instead of the pre-Botzinger complex telling the phrenic motor neurons to fire, the pacer delivers that signal directly. So Marissa here on the left is an example of how life-changing this can be. She spent 18 years dependent on nighttime mechanical ventilation. And after receiving this phrenic nerve pacers, she was able to sleep without a ventilator, which is fantastic. Improvement in life of quality. Now she was traveling, attending university, and she regained her independence. So it's really important. Physiology is very complicated. I appreciate that, but it's something that can really, really push modern day medicine. And without it, you really don't even stand a chance to come up with therapies. So here, even when the central rhythm generator fails, the peripheral pathway is intact and we can activate it artificially. So understanding the respiratory circuitry is exactly what made this therapy possible. All right, everyone, let's pause for a moment here. Take a breather, literally, before we jump into the high altitude section. Because of reading week, this part of the course unfortunately got squeezed in terms of the content. So we shortened lecture 4, and then we shifted the remaining material into today's session. So everything we're about to cover, hypoxia, altitude physiology, and chemoreflexes, is still core content for the course.